All right. Okay, so good afternoon, everyone, and thanks a lot for joining this month's new NeuroMeetup session. So this month, the NeuroMeetup talks are going to be dedicated to sensory perception during sleep. A very interesting topic because, as some of you may already have seen, the question of why we sleep made the cover of this month's issue of Science. So we are all very excited to hear more about the neuroscience of sleep. So today, our two speakers will be firstly Ida Boccalaro from the Inselspital on modulation of brain circuits for sensory processing during sleep states. And then Raquel Ortega from the Department of Physiology of the University of Bern on the processing of touch and pain during sleep. So just a quick reminder about how our NeuroMeetup sessions work. So they take place each month, usually the first Wednesday of the month at 5 p.m. And the novelty is that from now on, all our talks are going to be recorded and put online on our YouTube channel. So if you cannot attend or if you miss a talk, don't worry, you will be able to watch it offline later. And so you can also note right now that the next NeuroMeetup session is going to take place on Wednesday, the 1st of December. All right, so this being said, we can start with our first speaker, Ida Boccalaro from the Inselspital. So Ida, you obtained a bachelor's degree in biotechnology from the University of Perugia in Italy, and you wrote your thesis at the Department of Experimental Medicine in the section of Physiology and Biochemistry on the role of inflammasome in inflammatory pathologies. And then you obtained a master's degree in cellular and molecular biology at the University of Turin in Italy. And you obtained your master thesis, you wrote your master thesis at the Institute of Pharmacology and Toxicology from the University of Zurich on GABA receptor heterogeneity in striatal interneurons. So Ida, your talk is going to be dedicated to modulation of brain circuits for sensory processing during sleep states. You have 20 minutes to present your research, which will be followed by 10 minutes of Q&A, and the floor is yours.
Thank you very much. Hello, everybody. So I always like it to start the presentation with just a few features about sleep. So we know that when we are sleeping, there is this decreased sensitivity to an environment, a low arousal threshold, and lack of behavioral responses, so to be disconnection. But we also know that this disconnection cannot be total globally, because from an evolutionary point of view, we need to be aware from the surrounding in order to be able to understand if there is some dangerous sounds in the environment. And on this line, I'm just here putting some references of the possible mechanism of this decoupling during sleep. So the first hypothesis was the thalamic gating. So during sleep, the signal, the auditory signal in this case, was coming from the environment, was reaching the nuclei of the thalamus. But then at the level of the thalamus, there was this inhibition of the signal. And as we can see here from this paper from Adeline of 2001, they record that electrophysiological spikes in rats. And as we can see here, there was this decrease in the firing rate during slow wave sleep compared to wakefulness. So this paper wanted to point it out that there was this thalamic gating because the signals were not reaching the cortex as during wakefulness. Then the second hypothesis is the cortical gating. So the information, the signals during sleep is reaching the thalamus, but is also reaching the cortex. And at the level of the cortex, there is this decrease for the signals. But as we can see here in this first paper of Issa and Wang of 2008, they record that the auditory cortex of monkeys, and in gray here, we can see that the firing rate during the auditory stimulation was actually higher during slow wave sleep. And on the same line, also recording during non-REM and REM in rat auditory cortex also shows from this paper of NIR of 2015 an increase in the response during the audio stimuli. So the signal was reaching the cortex and also with an increased activity. Then the third hypothesis is the information gating. So the signal is going to the thalamus, from the thalamus is reaching the cortex. Then during sleep, since we have this thalamo-cortical oscillation, this slow oscillation can somehow modulate the evoked response to the stimuli from the environment. So here I pointed out just a few papers. The first is about non-REM sleep of Dang Vu of 2011. And here, as you can see, the evoked response upon auditory stimulation is completely decreased when the audio stimuli was in the same time of the spindle. Also in this paper of Dang Vu, what they saw is that the spindle rate was able to modulate the possibility to awake. So more spindle, less awakening. Furthermore, in this paper from Chabus, what they saw is that also the slow wave could somehow modulate the evoked response upon auditory stimulation. In fact, indeed, what they found was that the evoked response was higher when the stimuli was delivered on the negative to positive slope of the slow wave. Furthermore, even also during REM, we can see this paper in which when the stimuli, the auditory stimuli was delivered during the phasic REM, the evoked response was decreased. And same results also from Verle et al. Of 2007. And on this line, we go to my hypothesis, our hypothesis, that is that auditory-evoked slow wave travel across several brain areas, some of which are located in the medial dorsal thalamus that control auditory-evoked awakening. But why we wanted to do this project? First of all, because as I said, it's still an unknown mechanism and circuit of the auditory stimuli during sleep. Even if during wakefulness now it's already well known which are the nuclei that are activated upon auditory stimuli, during sleep is still unknown. Furthermore, there are more and more papers that are using the audio closed loop in order, using the auditory stimulation in order to increase the slow wave, but still the pathway during sleep is not so known. Also, the auditory stimulation could be a powerful tool for neuromodulation because it's not invasive in humans, as they did in this paper of 2021, in which they were able to target the slope of the slow wave with auditory stimulation. And they were able to decrease the centrotemporal spikes in this benign epilepsy childhood disease. And furthermore, as I've already said, there is lack of studies showing everything all together, all the slow wave and spindles can influence this auditory evoked awakening. So here we're going to my specific research questions that are, where do auditory evoked potential travel during sleep, and what is the thalamic contribution for auditory evoked awakening? As I said before, our hypothesis is that the central medial thalamus, since it's a hub for the sensory processing and also is a nuclei in which they take origins or the slow wave and the spindles, could have an important role in this sensory processing during sleep. How I did it, I used a mouse model in which I implanted multi-site tetrads in 4 different regions: the medial geniculate, the premonitory cortex, the hippocampus, and the central medial thalamus. Then I exposed the mice for 7 days at 9 different sounds. As we can see here, we have the evoked response that I obtained during the stimuli, auditory stimuli, during wakefulness, non-REM and REM. So first of all, as we can see, for all the targeted regions, I obtained an evoked response. Then, in line with the NIR study in the rats, auditory cortex, this evoked response was higher of amplitude during non-REM compared to wakefulness and REM. Also, we were we found it interesting because you can see that there is this super fast response between the first 50 milliseconds, in which the first one to be activated is the auditory cortex. And so we have this fast response of the central medial thalamus. And instead, then we have this second, a little bit later response that is higher in amplitude. So as a first response result, what we can see is, as I said, all the targeted regions have this ERP that is higher during NREM. And also we pointed out that there was this fast response of the central medial thalamus. Then we wanted to take a look more into the auditory-evoked awakening. So we split all the recording in events in which the animals will continue to stay sleeping, that you can see in blue in all the presentation, and instead events in which after the audio stimulation the animals were waking up, that you can see in red. As you can see here on the left, with this plotter pointing out is that when the auditory stimuli was coming on the negative to positive slope of the central medial thalamus, so when the central medial thalamus neurons are more excitable, the animals tended to wake up. Furthermore, we also wanted to take a look at the spindles, since all the literature is always wanted to understand the role of spindles in auditory-evoked awakening. And in line with the previous studies, we found that the events in which the animals will continue to sleep were characterized by a higher spindle rate, both in the EEG and in the central medial thalamus. So we have this slow wave influence on the auditory-evoked awakening, and also we have the first suggestion that could be also a possible role of CMT in this process. Then, in collaboration with Florence from the Athena Zovara lab, we also wanted to try a computational approach. So Flo trained her neural network with my electrophysiological recording in order to understand which region was the most important for the network in order to understand if the animals were waking up or not after the stimuli. And also the computational approach suggested the important role for the central medial thalamus. So also the computational approach confirmed the CMT role in auditory-evoked awakening. Then we wanted to look at the neural activity. So as you can see here, in the events in which the animals were not waking up, we had a decrease in the amplitude of the LFP for the central medial thalamus. We also have it a little bit for the auditory cortex, but it was not significant. So then we also tried to modulate this auditory-evoked awakening, silencing the central medial thalamus before, during, and after the auditory stimuli. And we were able to decrease the auditory-evoked awakening from non-REM. Instead, from REM, we were able to have this tendency just in the short latency, but there was not anymore in the long latency. So the opto-silencing of CMT was able to decrease this percentage of auditory-evoked awakening. Then we wanted also to understand what was happening when we had a neutral stimuli, so a safety stimuli, against a dangerous stimuli. This is why we wanted to run a fair conditioning protocol. So I recorded 2 hours of sleep in which I exposed the animals to just 2 sounds this time, the CS plus and the CS minus. And then on day 2, I paired just one of the sounds, so the CS plus, to a foot shock. On day 3, I checked if the animals were able to distinguish between the safety and the dangerous sounds. And then after this confirmation, I recorded 2 hours of sleep in which I was re-exposing the animals to these 2 sounds. As you can see here on the right, from non-REM, the percentage of awakening during the hours in baseline is the same for the CS minus and for the CS plus. Instead, on day 3, the animals were waking up from non-REM more for the CS plus. Didn't have the same results from REM. So the discriminative ability persisted during non-REM, but not during REM. So as to conclude, taken together, our results showed that the effect of the environmental auditory cues to induce awakening, in particular those associated with danger, strongly depends on the ongoing brain state, so slow waves and spindles, but also on the neural activity of CMT neurons. Thank you, Antoine, of course, but also the collaboration with Fla, all the members that did Zen and the IRC. Thank you for your attention. If there are any questions, I'm here. Also because I was super fast, as always. So please ask.
Yes, but it's nice because we have plenty of time for questions. So thanks a lot, Ida, for the very interesting talk. So I would just like to remind the audience that you can type in your question in the chat box so that I can send the questions to the speaker. So while waiting for the first question from the audience, I'm going to start with the first question on the convolutional neural network that you used to highlight the role of the CMT.
Yes.
Yeah. Okay. Is it possible to have some? We went quite fast on this slide. Is it possible to have some more details on this CNN, on the kind of data you fed it with? I guess you are feeding it with well, with the electrophysiological recording, and you train it to predict whether you are going to switch from NREM to wake or not. Is this right?
Yeah. But we try at the beginning, this is why I remember the first time that I presented this data set at my lab meeting, we were talking about prediction. Because of course, what we wanted to understand was if the neural network before the audio stimuli, based on the neural activity, was able to understand if the animals were, let's say, able to wake up, were more prominent to wake up or not. Then, so before time zero, so time zero is when the audio stimuli was coming, before we were not able to obtain some results, so the network was not able actually to understand. But then with this new one, so after the audio stimuli, we are able to. So I cannot talk about prediction because it's not, because the audio stimuli is there. But yeah, at the end, we just wanted to understand which regions were the more prominent to help the network to understand which was the transition, let's say.
Okay, and yet precisely about these areas that you analyze using the CNN. How do you translate the CNN into different areas? Do you look for the activity of specific cells or specific filters within the CNN?
Yeah. Here is in the spindle range. So from, if I remember well, from 10 to 14, something like this. So we filter everything in the spindle range. And then that was the activation per region that came out.
So you're feeding the spindle activity instead of the whole LFP?
Yes. At the beginning, all the LFP. But then, since we had the results with the spindles, we tried to be a little bit more specific. And so...
And do you get the same results with the whole LFP and the spindle activity,
yeah,
or is it different?
No, same results, but of course we had to remove the EMG because of course with the EMG he's able to understand every time, the network is able to understand every time that he is waking up or not, because the EMG is just makes sense.
You're having a startling response in the animals.
Yeah.
Okay.
We have another question from SF in the talk. So great talk, thank you. I am interested to know whether the intertrial variability of the evoked potential was the same in all conditions, wake, non-REM, REM, or larger in any of them?
We're the same. It's always a pseudo-random distributed, so with MATLAB, and the gap of silence between the different stimuli is always from 2 to, if I remember well, 11 seconds. So yes, okay. It's the same, just for the last part. So the free conditioning in which we just have two sounds. This was a different pseudo-random distribution, always with the gap, but this time we had just two sounds.
All right, thanks.
Maybe, sorry, maybe you misunderstood my question. I was asking whether the response was more variable. And the background to the question is there any indication that maybe the non-REM sleep has different periods of sensitivity, or is the response always the same?
But between different sounds or between sounds?
No,
okay.
The same sound has a different effect in the different stages. And within non-REM sleep, is the variability of the response from one sound to the next one larger?
Okay. No, I actually didn't look at it because unfortunately with the LFP, maybe with spikes is better, but with LFP, if you just have one event, it's really tricky because it's really noisy, the signal. This is why. But my outlook is to try to take also the spikes, and maybe with the spikes we could obtain, I got what you're meaning, like the first sounds that you are delivering to the mice are a different evoked response to the latest one, for example.
For example, or it's something like pre-REM, non-REM, as opposed to, so is it the non-REM after wake the same as the non-REM before REM, for example? So is there any differences
because with LFP I tried just to average everything in order to have like a more nicer response, let's say, but it's super interesting also because I have several days, so for sure there should be some differences. But for the awakening also, because now I'm just dividing everything wake, non-REM and REM, and the awakening from REM makes me so much trouble because it's deep sleep. So there is not really rules in order to understand what is happening. But from non-REM, I don't know, I think if there are, I don't know, 3 different sounds during non-REM, it could be, so it would be super interesting to actually look at it.
I'm looking forward to hear about it.
Okay.
Thanks.
Thank you.
And I had a question. For the optogenetic manipulation that you do in the CMT, right? So you're injecting in the CMT and then like inhibiting it, right? And the thing is that the CMT has a lot of connections with also prefrontal areas, right? So I was wondering, what is your take on this modulation of the awakening? Do you think that it's because the CMT is sort of preventing, like it's modifying the activity on prefrontal areas, or because it's kind of blocking the somehow the auditory processing?
For sure, the prefrontal cortex is always involved in everything. So for sure, there is also that modulation. But another outlook that I'm doing is to put together opto and free conditioning. And our idea is that instead of the prefrontal cortex, with the free conditioning, we would like to understand if the projection from the amygdala that is talking with the central medial thalamus, and the central medial thalamus talking with the amygdala, if we inhibit it, we are able to, for example, the animal are not able to discriminate between the dangers against the safety sounds. Because as you can see here, in all my injection, there is a huge projection to the amygdala. So it'll be super interesting. But of course, also with the prefrontal cortex. Also, it's several papers about prefrontal cortex and free conditioning, Matthias paper. So for sure, there is also there something.
And then also another question. So you said that you implant in the hippocampus, but then you don't show much about it. Can you explain what you found there?
Super honestly, at the beginning, we targeted the hippocampus just because we wanted to try to find a region in which we didn't have the evoked response. Because of course, at the beginning, we were super happy. We have an evoked response. But then we wanted at least to have one that was not flat, but something like this. And instead now, there is also several papers and also this collaboration with Sigur on humans that the auditory-evoked potential is traveling everywhere. So you will always have, even if small, an evoked response for the auditory stimulation. And
okay.
This is why at the beginning, actually.
And also, like, regarding the spindles, you said that when the spindles are high and in the EEG and also in the CMT, that you don't see this awakening response, right? So there's this protection. I was wondering, what do you see in the auditory cortex? And did you also check that on the hippocampus, if there are any spindles in the hippocampus?
Do you mean co-occurrence between spindle, auditory cortex, or just the spindle rate?
So if you find that the spindle rate is high in the CMT and also in the auditory cortex at the same time, in those stimuli where you don't have the awakening,
decrease of the awakening.
Or if you see that there is some mismatch between these two brain areas at some point.
I think I did it. No, I don't remember if I have here in the supplementary. Because this one, it was the spindle rate in the auditory cortex, as you can see here.
Yeah.
There were not just so many, but no, as you can see, actually, when there was the auditory, but wait, now I cannot see because I put the faces. Okay, baseline auditory stimuli. So no, I didn't, but I should, actually. I have the data. So it would be actually super interesting to check what's the spindle rate more at cortical level, and not just in the thalamic. Okay. So yeah, thank you. Very nice. I would look.
Oh, that would be interesting to look at.
Yeah. But also exactly to understand, I don't know, if there's see differences or something like this. Is true.
All right. So thanks a lot for answering all the questions. If we don't have any further questions, I suggest that we move on to the second speaker. So the second speaker will be Raquel Ortega on the processing of touch and pain during sleep. So Raquel obtained a bachelor and a master in human biology from the University of Barcelona, as well as a master in neuroscience from the University of Strasbourg. She did a master thesis on the antidepressant effect of lactate at the EPFL, and she's now a PhD student in the lab of Thomas Nevian at the Department of Physiology of the University of Bern. And Raquel, the floor is yours.
Thanks a lot, Camille. So in my PhD project, I try to understand the interactions between pain and sleep. And today I would like to show you the results that we have obtained in one of the sub-projects in which I have been looking at the processing of pain and touch during sleep. And this is because, as very well Ida has said before, for many years it was believed that when we sleep, we are completely disconnected from the environment. We cannot feel anything around us. And that was because the peripheral information could reach the thalamus, but then could not be transferred to the cortex to be processed. So we could not know what is going on around us. But as Ida has shown in her project, and as many others are showing, this is not entirely true. In this study, which was one of the first ones showing that, they implanted rats in the auditory cortex, they placed some tones, and they saw that actually the neurons in the auditory cortex were responding to these tones. And when they averaged these responses, they saw that actually they were not so different from the different arousal states, meaning wake, non-REM, and REM. And as more that we know about this processing, as Ida has shown, we see that there are actually more subtle differences between the processing in these different arousal states. And over the years, we also have seen that humans can understand words when we're asleep, and we can understand them and we can classify it. And this is not exclusive for auditory stimulations, but we have also seen that in the visual information. But what about touch and what about pain? We don't know much about these senses. And this is because they're actually quite challenging to study and there are some technical difficulties. And among them, we know that the pain experience is rather complex and it has multiple dimensions. So the pain can be divided, for instance, in sensation, affection, and cognition. And in sensation, we have the characteristics of pain. That is what is known as the sensory discriminative component of pain. Then there is the affective, motivational component of pain that processes all the negative emotions that come with the experience of pain, as well as the motivations to react to that. And then we also have the interpretation of pain, this third dimension. And today we'll talk about this sensation and affection dimensions of the experience of pain. From anatomical studies, we know that these two components of pain are processed by different thalamo-cortical pathways. And the natural pain pathway that includes the VPL in the thalamus and the somatosensory cortex tells us about the sensory component, while the medial pain pathway that includes the MD in the thalamus and the anterior cingulate cortex processes the emotions that accompany the experience of pain. So to study how we process pain during sleep, we have implanted mice at the very end of these two pain pathways. That means in the ACC to study the emotions of pain, and in the somatosensory cortex to study the sensory discriminative component. More precisely, we have implanted the somatosensory cortex of the hind limb. And then when the animals recover, we did the recordings in which we put the animals on a grid, and then we poke the hind paw with something that was either non-noxious or non-painful, or something that was noxious, painful. And one recording session would look like this. Each vertical line is a stimulation. And the interesting part of this of this protocol is that each stimulus will fall into a different arousal state, right? So they will be done either in wake, in REM, or in non-REM. And today we'll show you the results that we have obtained from the stimuli that were done in non-REM sleep. The first thing that we observe is that these stimuli evoke different responses to these animals. So the animals either did not react at all, or withdraw the paw only, or woke up only, or woke up and withdraw the paw. And you can see that the proportions of these two different different behavioral reactions are different depending on the stimulation type that we did. And I would like to start showing the results that we had for this stimuli in which the animals did not show any reaction. And this is why the first question that we ask is, do touch and pain reach the cortex during sleep? And to avoid having contamination of waking up processes or motor responses, then we chose to have a look at this stimuli first. And here are the voltage recordings of the populations of neurons in the somatosensory cortex and in the ACC. And you can see that after the stimulation onset, we have these evoke response potentials here that are present in both cortices, in the somatosensory cortex and also in the ACC. And when we looked at some characteristics of these ERPs, so meaning the voltage at the trough and the latency to the trough, because the trough is something, it's a characteristic that it's very consistent throughout trials, we saw that actually the voltage at the trough did not differ from the somatosensory or the ACC, and also, but there was no difference much difference between the painful and not painful. Now, when we look at the latency to the trough, we saw that the somatosensory cortex was much faster than the ACC. However, if you look carefully at these evoked response potentials, you see that the dynamics are very different between the somatosensory cortex and the ACC. Being the somatosensory cortex the first one to react in both cases, and then the ACC coming afterwards. So we're doing now a more complex analysis to look at all these dynamics. But we can also look at these voltage potentials from the frequency perspective. And to do that, we took these signals and we look at the different frequency components of these signals, because the different frequencies gives us different information. And this is what you can see here. These are spectrograms in which on the y-axis you have the different frequencies, which are the different frequencies components of the previous signal. And on the x-axis you have the time. And the first observation that we can make here, and that is very striking, is that after the stimulation onset on the somatosensory cortex, there is this increase in the higher frequencies oscillation, so the frequencies above 30 hertz. And this increase is very prominent for those painful and non-painful stimuli. When we see this decrease in the higher frequencies, it means that there is some cognitive processing going on. So we can assume that the sensory discriminative aspects of pain are being processed during sleep. Now, when we look at the activity of the ACC, we also see that especially for the painful stimuli, the noxious stimuli, there is an increase in the higher frequencies, and this increase is not so strong for those stimuli that were performed in that were not painful. And this tells us that these painful stimulus, they also have the emotional component being processed in the ACC. So to have a more quantitative approach of these changes, we have focused into these two frequency ranges. The higher frequencies are called gamma, and we have also looked at the sigma frequencies because it seems that there is a very prominent decrease of the power of these frequencies. All right, so when we look at the gamma frequencies, we see that for those stimuli that were painful, there is a sudden increase at the stimulation onset in both the ACC and the somatosensory cortex. And here I'm showing you those trials that did not have a behavioral response that are in black, and those trials that had a behavioral response that are in red. And for the noxious stimuli, it doesn't matter what kind of stimulus we're doing, we see this increase. Now, when we look at the non-painful stimulation, this changes a bit, right? So those trials with behavioral response have a stronger activation of the gamma frequency compared to those without behavioral response. But in the somatosensory cortex, we still see that there is a prominent increase for both types of trials. But in the ACC, this is not the case for the non-noxious stimulation. So it means that the emotional component for those touch stimuli is not so strong as for those painful stimulations. Then when we look at the sigma range, we see that there is this decrease in the power. Let's have a look at the somatosensory cortex. In those trials without behavioral response, and this power doesn't go back to the power at the baseline time. In the case of the trials with behavioral response, they don't go to the original power before the stimulation onset, but we see that there is a better recovery. And we see that there are also some differences between both cortical areas and also between both types of stimulation, what indicates that there is a different processing of these stimuli in these two cortical areas. And as Ida has illustrated very nicely in her presentation, we know that the lower frequencies have a protective effect to sleep, right? So what we did was to select these three frequencies and look at what happens before the stimulation onset. And here we have something very interesting. So again, black line trials without response, red line trial with response, any response. And we see that before the stimulation onset for these 3 frequency ranges, the power is higher if there is no behavioral response. So it's kind of some sort of protection for the sleep. But now, when we look at the somatosensory cortex, we don't see this difference anymore for the delta nor the theta range, but we see them in the sigma range, which goes in line with what Ida has shown before, that the sigma range is a proxy for the sleep spindles. So in these cases, we assume that there had some more spindles before the stimulation onset, and therefore the animals are protected from doing a behavior. So after seeing that, we decided to take those trials that had a response and divide them into the different behavioral responses that I've shown you before that we have. And this is what we've seen. So, so that it's clear, the first bar, it's those trials that have no response. The second bar is when the animals wake up only. The third bar is when the animals withdraw the paw only. And the fourth bar are the trials where the animals wake up and withdraw the paw. And when we look at the power of delta in the ACC for those stimuli that were non-painful, we see that when this power is high, there is this protection, and it comes out significant for no response versus awakening only. And we see that there is a trend here that doesn't come out significant. But then when we look at the theta range, we see that when the power of theta is high, there is a protection against awakening, but not withdrawal of the paw. And when we look at sigma, we see that the higher the sigma is, the more protection there is. And there is protection against awakening and against paw withdrawal. Now, when we look at the noxious stimulation, we see that there is some sort of gain of protection from this delta power. When we look at theta, it seems that there is a shift. There is no protection anymore from awakening, but from paw withdrawal. And when we look at sigma, we see that the protection of sleep is being kept, right? The higher the power of sigma, the less reaction that the animal has. When we look at the somatosensory cortex, we see this. We see that there is a protection from waking up, the higher that the power of these 3 frequencies are. But when we go to the noxious stimuli, we see that, well, this protection is maintained for sigma, but there changes for the delta and the theta. So then we see that it's a bit complicated to interpret that, right? Because the protection sort of depends on the strength of the stimuli and the frequency range and the cortical area that we're looking at. So what we decided to do now is to do a ratio of these frequencies, of the low frequencies against low frequencies and low frequencies against higher frequencies. And we didn't find anything for the ratio between delta theta or delta sigma, but what we found, it was also very interesting, is that there are differences for the ratios of low frequencies versus high frequencies. So let me show you this. Here are the ratios for this non-painful stimuli in the ACC. And this is gamma. So gamma, if you remember, those are the frequencies between 30 Hertz and 100 Hertz. And this is the ratio between delta and gamma, theta and gamma, and sigma and gamma. And what we can see is that when this ratio is high of either these 3 low frequencies versus gamma, there is a protection from awakening. That's what you can see here. So the green are trials where the animals wake up, and the blue are trials where the animals do not wake up. So there is a protection from awakening, but there is no protection for withdrawal. If we look now at the somatosensory cortex, we see that, well, we actually see that there is not this protection anymore for neither of these three ratios, right? Now let's have a look at what happens with the painful stimuli. So with the painful stimuli, we lose the protection that we have with this delta gamma ratio and the theta gamma ratio. But it seems that the signal, the sigma, still keeps its protective effect, even when the stimulation is very strong. And now when we look at the somatosensory cortex, we see that there is some protection for the theta gamma ratio, and the sigma, again, it's so strong that it protects from any kind of behavioral reaction, right? So these results, what they're telling us is that it seems that the state of the ACC and the somatosensory cortex are very important to define what is the behavioral reactions that the animals will have. And it seems that the effect of the state on the ACC previous to the stimulation onset is different to the state, to the effect of the state of the somatosensory cortex. So it seems that there is some sort of interaction between the ACC and the S1 that will define the type of behavioral reaction that we will see. And this is something that we're working on today. I will not show you more than this, but the take-home message that I would like you to have is that we have shown for the first time in naturally sleeping animals, the touch and pain can reach the somatosensory cortex and the ACC during sleep. And that we see also that there is this processing in these two cortical areas, indicating that the sensory discriminative and the affective motivational components of pain are being processed during non-REM sleep. I have also shown you that the high power of the low frequencies have a protective effect on sleep, and that the state of the ACC and the somatosensory cortex before the stimulation onset defines the type of the behavioral response that we're going to see. And with that, I would like to acknowledge my colleagues, and especially Marco Renard, who has helped a lot with the experiments, and also the funding source of this project, the Interfaculty Research Cooperation, Decoding Sleep. And I would also like to thank you for your attention, and if you have any question, I would be more than happy to answer them.
All right, thanks a lot, Raquel. Thanks a lot for the very interesting talk. So once again, I invite members of the audience to write down questions they might have in the chat box. While waiting for them, I just have two questions. I was really excited by the result you've shown about how the neural states in S1 and ACC predict the behavioral response of the animal. So maybe the first question I have is, so far the results you've shown are a posteriori analysis. That is to say, you observe the power in different frequency bands after the response, the behavioral response has been recorded, and you see whether there is a significant difference between different conditions. Did you try to, I don't know, to train a classifier or a decoder to see whether such a classifier would be able to predict in advance the response, the behavioral response of the animal, just given like the state of S1 and the ACC prior to the stimulus?
So that's something that it's in the pipeline. That's something that I want to do, because I think that there is a lot of potential that by feeding this information of these two cortical areas, we can predict what is the behavioral outcome that we will see in the animals. Indeed, I mean, we haven't started that yet, but it's definitely something that we want to do. Yes.
Yeah, it would be a nice addition to your results, and it's well, I'm a theoretician, so it's always cool to have a little bit of machine learning on top of experiments. And also the second question, that might be a very naive question. I'm not an experimentalist and let alone a sleep researcher, but would it be possible to artificially modify the activity so as to increase or decrease the power in different frequency bands, so as to modify the response of the animal to a stimulus? I'm asking the question because I wake up very easily whenever there is very small noise, and would it be possible to artificially hack my brain so as to prevent this?
Mm-hmm. So we're working currently on that. That's Margot Renard, who is now setting up all the pipeline for these experiments. And so that's the idea, to modify the activity of these two cortical areas to see whether we can see a change in the behavioral reaction of these animals at the time of the stimulation. And we plan to start these experiments this month with the implantations, and I hope to have some results already in a couple of months. But yeah, definitely. So that would also open the door to possible treatments for feeling less pain, right? Because that would also modify, that has the potential to modify the sensory threshold of the animals. Exactly.
Yes, definitely very exciting. Okay, do we have any other questions? Maybe if you could let us know a little bit more about this interfaculty research cooperation. I never heard of it.
Mm-hmm. Yeah, so this is a very nice cooperation that started at from the University of Bern, in which there are multiple labs working together about the topic of sleep. And we are trying to look at sleep from different perspectives. And for instance, so Ida is also in this IRC, and we're in the basic research part in which we study this sensory perception. There are also other groups doing similar studies in humans, and there is also the part of computational to create closed loop stimulation during sleep, and to create models of sleep and the roles of non-REM and REM in memory, for instance. If you want more information, I have left here the website, so you can check with more detail what are we doing and what are the groups that are involved in it.
All right, thanks. Do we have a question from Jakub. Thank you very much for the nice talk. I wanted to ask about the frequency of stimulation. Is it somehow controlled or random?
Yeah, so the stimuli that we perform are random. We don't want the stimuli to always have the same interval. And this is because when we have these oscillations in the brain, we want to fall into different parts of the oscillation. We don't always want to fall into the same phase, right? Because then that would bias our results. So the stimuli that we perform, they have an intertrial interval that goes from 20 seconds to 20 minutes. So like this, we make really sure that we're not always falling into the same phase of the different oscillations.
Do we have any more questions from the audience? All right, so I guess that's the end of this month's session. Thanks a lot, Raquel. Thanks a lot, Ida, for the very interesting talks. It was very exciting. All right, so thanks a lot for the audience for attending. And you are all welcome to attend our next session that will take place next month on Wednesday, the 1st of December. The speakers and topic are still to be announced, but we hope you will attend as well. All right, so thanks a lot again to both speakers. Thanks a lot to the audience, and we hopefully will see you next month. Thanks a lot.
Thank you, Raquel. Thank you, Camille. Bye.
Bye, everyone.
Bye.